All right, well, hello everyone. My name is Korowinski. I am a first-year PhD student here in the kinesiology program, alongside my mentors, Dr. Vakuda and Dr. Stout in the Power Lab. Before I introduce our next guest, I just want to remind everyone that our conference is sponsored by Keg Social. Whether you're in the mood to let loose with your friends, create amazing memories with your family, or bond with your team, Keg Social is the ultimate destination to amp up your night out. There's a location at Waterford Lakes, not too far from here, so we hope to see you at Keg Social soon. And with that, I am truly honored to introduce our next speaker, Dr. Mike Roberts. Dr. Roberts completed his PhD at the University of Oklahoma, followed by a postdoc at the University of Missouri, under the mentorship of Dr. Frank Booth. Dr. Roberts here is an Auburn University alumni professor in the School of Kinesiology, where he serves as the director for the Nutrable Applied and Molecular Physiology Lab. Dr. Roberts' lab utilizes cell cultures, rodent and human models to examine how nutrition and/or exercise affects the cellular markers in humans and whole body physiology. He currently has over 200 publications in several preeminent physiology and nutrition journals. He serves as senior editor for various physiology journals, and he's given numerous lectures at the regional, national, and international levels. And so the title of his lecture here today is The Past, Present, and Future of Skeletal Muscle Hypertrophy Research. So if you all can please join me in welcoming Dr. Mike Roberts to UCF.
Thank you, Corey, for that nice introduction. Thank you all in attendance, and I want to thank my friend Matt Stock for the invite, as well as Jeff and Joel and UCF. So let's see if I don't mess up this clicker. Again, Dr. Stock, Jeff, David, Joel. Apologies, Joel, for not having your name there, as well as Dr. Herda. It's good to see old friends in the crowd. I need to also thank all of the work that my students do at Auburn University. I am just conveying all of their hard work that they've done in the lab over the years. And then I've had the opportunity to work with a lot of very interesting people, one of which I'm going to talk about today, a paper I did with Karen. But in particular, I'll be discussing some data we've worked with Troy Hornberger over, as well as Gustavo Nader and John McCarthy and Stu Phillips. And then financial contributors for data in the lecture mainly is through Peanut Institute with some of our training studies. I also got internal funding from Auburn University as well. And then we've worked with some industry partners such as BNRG, Nutrabolt, and Lockwood, Dr. Chris Lockwood and his company. So yes, we do a lot of industry-sponsored research. In particular, we try to form strategic partnerships to determine nutritional supplements or whey protein type products or amino acids that affect skeletal muscle hypertrophy. I've also received internal funding for some of our research as well. I am a paid consultant for certain companies, mainly white paper educational materials. But in regards to the data that I'm going to present today, I don't have any conflicts to disclose. And BioRender says, make sure you cite us. I used a lot of BioRender to make diagrams, so I just want to make sure I do that. All right, to the brass tacks. So today's lecture is covering sort of skeletal muscle hypertrophy. How relevant is that topic right now? Well, if anyone uses Google Trends, it's pretty nifty because you can see some of the data from Google and what things are being searched. And what's interesting about this is since 2004, worldwide, there is an upward trajectory for people that are looking for the term muscle hypertrophy. And I don't think we get data on Google from China or Russia, but what I can tell you is it is a large interest, especially some of the historical Russian research in that particular topic. Additional food for thought here. I did some comparisons, and this is kind of neat because you can sort of stack different search terms on Google Trends. And if you look from 2004 over time, hypertrophy training has taken over cardiovascular training, right? So there's two ways to view these trends. One of which is the meatheads are finally winning the fitness wars, right? And I say in parentheses, NSCA celebrates, ACSM sheds a tear, little tongue in cheek there. But the other thing I think, which is a little bit more responsible for these trends, is, as all of us know, we are living in a population that is continuing to age, and we're going to have a lot more people that are of older age here in the next 30 years. This is one of the trends that I found from the literature, and they say by the year 2100, 1 out of 4 people are going to be 65 years or older. And so knowing that data, I went back to Google Trends, and I think what's interesting is the trend for hypertrophy training is on an upward trajectory. I discussed that. And the trend for the search term muscle loss is also on that same upward trajectory. So I think there's a lot more people that are concerned that with aging, there is this accompanied muscle loss. I know this is correlation, as Dr. Goodpaster said, correlation doesn't mean causation. But what I can tell you is this: we do a lot of training studies that involve resistance training, and we've done a few now with older participants, and we get a huge interest from the local community. They want to sign up not because of the paycheck, but they want to actually sign up because they're getting good quality supervised resistance training. All right, so striking while the iron's hot. I phone call a great professional mentor of mine. Her name is Karen Esser. She's at UF. And I said, Karen, look, you did a lot of great research in this area. In particular, she has a 1999 paper showing that mTOR signaling is operative when rat muscle contracts under load. And I said, it'd be great to do a historical piece on skeletal muscle hypertrophy and make it this really nice scoping review on mechanisms associated therein. And she said, not my area no more, no thanks. So if you know Karen, I know Brett, you know Karen, she can be very quick and direct. And so I did my smooth talking with Karen. I said, Karen, just think about it. You have great data. You were, a pioneer in this field. We can make it a historical piece in terms of like talking about the history of muscle hypertrophy research. And then she finally said, okay, fine. I said, all right, cool. So after 80 pages of content, and that excludes references, 225 emails I counted for this conference. I wanted to document that. And this took 1.5 years of work. We put out a review in Physiological Reviews that was published late last year. And myself and Karen, we got a lot of phenomenal people to contribute to this article. So Dr. Stock wanted me to really base the lecture on this review and some of the main highlights. And here are the main highlights. First, it's way too long. So don't take the time to read the entire thing, right? I can tell you that because I've read it about 103 times. I think the most interesting part and the funnest thing that I was involved with when we wrote this review was the historical section. And I got to give a shout out to Frank, as well as Gustavo, Nader, and Karen. And I want to ponder on this for a second. I have some fun trivia here, but can anybody in the audience, do they know what the first exercise skeletal muscle hypertrophy paper was? What year was that published? What group? Any guesses? 1893. You were so close. Did you know that? Yep. It was 1895. Very good. And what these authors documented, by the way, this paper is in German, and even when you use Google Translate, it is entirely difficult to read. The author was Morpurgo, and I can't find the first name of this person. It's that old, right? So they saw that 2 months of treadmill training in dogs increased myofiber diameters, but that sort of training did not increase myofiber numbers. So there was no hyperplasia. The other fascinating thing about this is that we're talking about, cardiovascular exercise in these animals causing skeletal muscle hypertrophy. Any guesses as to when the first muscle molecular paper involving resistance training was published? Any guesses? I'm going to look at you now. You said 1893. So this was Jeff Goldsmake, and this was in 1964, and this was in mice. right? So how did they resistance train mice? Well, they did this for 25 days. They set up a really slick pulley system so that when the mice wanted to eat, right, they had to pull on this pulley lever and it was resistance loaded. And what they showed was this increase in myofiber diameters. And this is kind of what's interesting. This is sort of the paradigm that they develop, but they say, hey, look, when you have myofiber hypertrophy with resistance training or mechanical overload, that's due to this increase in the size of pre-existing myofibrils within the myofibers. I want to come back to this, so save your receipts on that particular topic. All right. I just want to say some other things related to the history of this field, but Goldspink published this in Mice in 1964. The first human study utilizing biopsies looking at some of these similar measures was published 15 years later. Stefano Schiaffano published a paper in 1976 showing that satellite cells fuse to pre-existing fibers with mechanical overload, and the first human study was done 23 years later, excuse me. And then lastly, Sue Bodine published that mTOR signaling was very important for muscle protein synthesis in response to mechanical overload, and this was replicated in humans 5 years later, right? So I think one thing you can sort of take away from this lecture is that, hey, look, don't be this person. Yeah, well, you know, it's not important because it was done in rodents. One thing I've learned to appreciate is that the rodent researchers, they really do push the field, especially as it relates to skeletal muscle hypertrophy. And by the way, this picture comes from a baseball card. I don't know if any of you collect. I collect baseball cards. So, and then the other thing too is when you devote yourself to a particular topic that you're researching, I encourage you to go way back, right? Be a student of the history of that topic. Thinking about Joel Kramer, I remember Moritani and DeVries, 1979, and EMG, and you know, you come to appreciate how much thought was put into those older data sets, and they were sophisticated techniques by then, back then as well. The third thing from our article was that a lot of folks have examined overload hypertrophy, and really there's been 3 mechanisms that have been highly studied in relation to skeletal muscle hypertrophy. Do we have any guesses on those top 3 mechanisms? And this is a graph sort of depicting the number of articles published on mechanisms associated with hypertrophy. So what you're seeing, numbers 1, 2, 3 there, are the top 3 topics over the past 5 years, where number 3 has, 15 articles mentioning this particular mechanism. Any guesses? I'm thinking of molecular mechanisms. Yeah, that's a good guess, Brett. So the first would be ribosome. Yeah, exactly. That's kind of a common intersect, molecular marker. So number 3 is ribosome biogenesis. Number 2 is the involvement of satellite cells. And then number 1 would be mTORC1 signaling and muscle protein synthesis. Now, there's been a lot more mechanisms mentioned over the past 5 years. I'm not going to go into those today, but I will mention one thing, which is genetics. We'll talk a little bit about that. So, the consensus on mTORC1 and muscle protein synthesis, I think all of us agreed when writing this review article that certainly it's important. And the story goes, when you do one bout of exercise and you take biopsies post-exercise, you'll see this increase in mTORC1 activation, and that's going to lead to translation initiation, which is synonymous with an increase in muscle protein synthesis. That's practically undebatable. I think where there's a lot of nuance is upstream mechanisms. So whether you're talking about IGF-1 signaling and the upregulation of IGF-1 gene expression to weightlifting and whether or not that's important for skeletal muscle hypertrophy can be debated. Another thing that's been pontificated is mechanotransduction. Has anyone in the crowd heard of mechanotransduction? And focal adhesion kinase phosphorylation, again, that's debatable. These stretch-activated calcium or ion channels have been implicated in activating this mTORC1, but again, a lot of that data is in rodents or in vitro. And then finally, Troy Hornberger, he's really made they have performed a series of studies showing that this little protein DGK zeta in the muscle cell is activated when you contract muscle. I think there's a lot of promise there. But the main takeaway is, hey, look, we know mTORC1 and muscle protein synthesis are important for mechanical overload-induced hypertrophy, but really there's a lot of conflicting evidence with these upstream mechanisms, and there's a lot of future opportunity for research to be done in this area. So sort of candidate number 2 in terms of mechanisms is the involvement of satellite cells with skeletal muscle hypertrophy, as well as an increase in myonuclear number, which is MN number up there when you weight train. And so how this goes is if you do weeks to months of resistance training and you take a pre and a post-intervention biopsy, you can do some histology using that sample where you cut the muscle tissue in cross sections. So you're looking at these long muscle cells in this transverse orientation. And what you'll find, this is from our laboratory, if you do fiber type specific staining, I apologize for not having a pointer, but type 1 fibers and type 2 fibers hypertrophy, we seem to think that type 2 fibers hypertrophy more. And you'll see this increase in satellite cell number as well as myonuclear number with resistance training. So people seem to think that this is an important adaptation. One study to really show this was from the Brazilian group that Carlos Ribosome is a PI of, and they did a meta-analysis that involved 27 studies, including 903 participants. And they said, hey, look, you can have this increase in fiber hypertrophy and myonuclear number coming from this satellite cell-mediated fusion to pre-existing myofibers. However, you have a really robust increase in myonuclear number with myofiber hypertrophy that exceeds 22% with resistance training. Another study that illustrates this concept well comes from Marcus Baumann's group, and this was back in the mid-2000s. But what this group did was they had higher and lower hypertrophic responders to resistance training, and they looked at the histological characteristics of these individuals prior to training. What they found was higher responders had more satellite cells prior to a period of resistance training, and then after 4 months of training, right, the lower responders saw less than a 5% increase in the size of myofibers, whereas the higher responders saw this robust hypertrophy, like a 60% increase. And that was associated with more satellite cells as well as a greater myonuclear accretion. All of this to say that we think that satellite cells are a really important contributor to skeletal muscle hypertrophy, and I think that's become almost undebatable at this point. Now, while we had our little kumbaya moments in writing this review about mTORC1, we had some diverging opinions in terms of the importance of satellite cells, and that's because we had some rodent-based researchers, such as John McCarthy and Karen Esser, contributing to the piece, and then we had human folks like myself, Carlos, and Stu Phillips. And one paper that was published in 2011 was this one by John and Karen and Charlotte Peterson, showing that when they use a genetic mouse model to ablate satellite cells and then they perform mechanical overload, they can still have skeletal muscle hypertrophy in these rodents. Now that has since been challenged by Christian Gunderson's group and other subsequent studies from the Kentucky group. But regardless, there's still a little bit of debate in terms of the importance or the centricity of satellite cells to skeletal muscle hypertrophy. And the other thing to consider, and I think this is a very innovative way to look at satellite cells, but we have this fusion role, right? Where when I lift weights, I'm gonna have satellite cells in my muscle bed proliferate, and then some of those will fuse to my pre-existing fibers and contribute to myofiber hypertrophy. So that would be the fusion role. But now there's talk about non-fusion roles, where when you lift weights, your satellite cells can secrete extracellular vesicles containing cargo, such as microRNA or peptides or other bioactive molecules that we haven't even identified. And with MoTrPAC, maybe we get a glimpse of some of that. But these extracellular vesicles that are secreted from satellite cells in response to lifting weights can communicate with fibroblasts, with other cell types that are in the extracellular matrix, and that can influence the tissue response to resistance training. So I think that's a very ripe area of research, especially in humans. And I say that because the only data that really supports that comes from Chris Fry's group, as well as John McCarthy. And I think we need to head there with our human research. The third topic, the third candidate mechanism that's been highly sort of mentioned in the context of skeletal muscle hypertrophy has been ribosome biogenesis. When you weight train, and again, the story goes, if you do 12 or so weeks of resistance training and you take a pre and a post-study biopsy, you'll see this increase in ribosome content. Now, this comes from our laboratory. I apologize, it's a little small, but what I'm showing you from in our hands is lower responders based on VL thickness changes after 12 weeks of resistance training, where the open bars were the low responders, and then the gray bars were the higher responders. And to Brett's point, I don't say non-responders, because what I'm not showing you is strength changes, and all participants practically got stronger, right? This is ribosome content when you look at the lower and the higher responders. And what you'll notice is that muscle ribosome content significantly increased, whereas that was in the high responders, whereas in the low responders, we got a non-significant numerical increase. So that's just an example showing you that ribosome biogenesis is associated with the hypertrophic response to resistance training. I think there's other interesting avenues that we can take down this path. One that Tom Shalhoub and John McCarthy have mentioned is ribosome specialization. So for those that are familiar with ribosomes, they contain ribosome RNA as well as around 80 ribosomal proteins. And ribosome specialization would be the post-translational modifications to those ribosomal proteins and whether or not those modifications make a difference with the hypertrophic response. I don't have time to get into the details because of time, but I think that's a ripe area. All right, so I've sort of given you a glimpse as to the three really hot topic mechanisms that have been associated with skeletal muscle hypertrophy. Where do we go from here? Well, Dr. Spock has us covered. I think there's a lot of novel and cool areas to explore with skeletal muscle hypertrophy. And so I'm going to just discuss a couple of these topics. The first is genetics. I think this is an underdiscovered frontier. I found this on bodybuilding.com. I think now we have a room full of younger people that don't even know what that is. Jeff laughs because I think Jeff and I have both written for them, and I think they've gone out of existence. But I found a thread back from 2004, and the thread title was Good Genetics, Bad Genetics, How Can You Tell? All right. Now we have Shut and Squat say, hi, I was wondering, well, how can someone ever tell who has good genetics? I mean, not too many of us that lift have been gifted. Loved by many, hated by few says, if you and somebody else are lifting natural, I guess meaning not taking steroids, and y'all started lifting from the same date, and lift equally or almost equally, if you have a bigger look, pardon the misspelling, I pulled that copy-paste, it means you got good genetics. All right. So I don't know if y'all caught the content of what loved by many, hated by few said there, but what he's saying is if you take two people and they train the same way for 12 weeks in the weight room and somebody gets larger than the other person, then they probably have good genetics. Now, if I polled this room and I said, hey, what's the veracity of this statement? I bet you a lot of us would say, yeah, I mean, that's pretty reasonable. We've said that for years, right? Some people have good genetics, some people don't have good genetics. Then if I ask, well, which polymorphisms are highly predictive of the hypertrophic response, I could probably hear a cricket in the back of the room, right? Now, if there's a genetics guru in the audience that stomps on the crickets, and says, hey, Mike, duh, I mean, we have alpha-actinin 3 polymorphisms, in particular that RX genotype, right? There's myostatin variants that have been documented in literature. There's IGF-1 variants, right? And now there's some evidence that the variation in our DNA copy number may be predictive of muscle hypertrophy in response to resistance training. Well, we did a study with Stu Phillips' group looking at over 315,000 polymorphisms in 109 men with 10 to 12 weeks of resistance training. And we ruled these 3 out, okay? And this was, this pretty highly sophisticated statistical approach. We didn't have anything on our array that tested for our DNA copy number. And so we were very interested in this topic. Here's the deal with our DNA copy number. I'm going to give you a couple of visuals here. The rDNA gene is used to make ribosomes, okay? And I already mentioned that ribosomes are critically important because I may have mentioned this, but I will mention it. Ribosomes catalyze muscle protein synthesis in muscle cells, and we know muscle protein synthesis is important, right? You can have, unlike most genes, several copies of this rDNA gene. They localize to the centromeric regions of chromosomes, and one person versus another can have hundreds of copies of differences in that particular rDNA gene. And so with that, I think a good hypothetical scenario could be, let's say we take somebody that has a low rDNA copy number, we take somebody that has a high rDNA copy number, we have them resistance train, and perhaps someone that has a low rDNA copy number doesn't make as many ribosomes with resistance training and therefore has less skeletal muscle hypertrophy versus the other person that has more rDNA copy numbers. Does that make sense? Kind of stumbled through that, okay? But I think this is a good hypothetical scenario, and there was a paper from Kentucky, Vondre put this out, that really supports this model. Now, the limitation was there were only 7 humans, and it was an acute study saying people with more rDNA copy numbers can have a greater increase in ribosome biogenesis in response to 1 bout of training. But nonetheless, it supported the paradigm that I just mentioned. All right. Well, we had a lot of tissue banked, and we said we need to tackle this question. So we had 82 participants, 53 being men and 29 being women, that completed 10 to 12 weeks of resistance training in our laboratory. Now, this is an important note I'm not going to show you bar graphs on, but both genders showed skeletal muscle hypertrophy at both the whole body level as well as ultrasound VL thickness and mean fiber CSA. So if the model of rDNA copy number held true, what we had hoped to see is this. On your y-axis, right, the change in hypertrophic score, so the change in whole body lean mass, the change in VL cross-sectional area or VL thickness, we would hope to see a positive association with rDNA copy number that looked like that, right? And statistically speaking, we'd hope to see something like this, a highly significant correlation. All right, this is what we saw with the change in whole body lean mass according to the DEXA and rDNA copy number after 10 to 12 weeks of resistance training. This is what we saw with the change in VL thickness according to ultrasound and rDNA copy number. And then this is what we saw with the change in mean fiber CSA and rDNA copy number. So we're not seeing anything there. We did other molecular techniques looking at ribosome biogenesis markers, and those did seem to show an association with hypertrophy. So what we think is happening is, hey, look, if you have low rDNA copy number, and this is a genetic phenomenon, you shouldn't worry because when you resistance train, it does not impair the hypertrophic response, okay? Josh Godwin, and by the way, Josh led this project. He's in my lab. He's going to go postdoc with Gustavo Nader, but we got Gustavo on board because Gustavo is the person that practically discovered, ribosome biogenesis with skeletal muscle when he was in Karen Esser's lab. He gave us a neat little tidbit, and he said, hey, why don't you take human muscle cells, culture them in Petri dishes, and give them a compound called BPA, or bisphenol A, cheers, right? There is a paper showing that reduces rDNA copy number in these cells. So sure enough, we did it, and what you're looking at are those data. When we put BPA on myotubes in vitro, it reduces rDNA copy number. With that, we thought, well, if we're reducing rDNA copy number, we should see atrophy in these myotubes. We did not, okay? And in addition, when we gave IGF-1, which stimulates anabolism, to control cells or cells treated with BPA, we saw a similar magnitude of myotube hypertrophy, right? So are genetics a big deal? Of course. I would be dumb to say that genetics don't play a role in the exercise response. We have the heritage study, right, showing that genetics are indeed a factor, and that can explain some of the variance to 20 weeks of aerobic training. But I think we need more and higher quality research. There are different areas. For instance, I've seen papers looking at novel genetic polymorphisms with IBD, inflammatory bowel disease, where they're using deep sequencing to identify these novel polymorphisms. I think we need to do that with resistance training studies. That's where we are at the technology level. We can do sequencing now, and we also need big computing to look at the role of combinations of different polymorphisms and whether or not that predicts the hypertrophic stimulus. I guess the only food for thought or the only takeaway here is just don't use genetics as gospel, because again, I've been guilty of saying, hey, genetics plays a role in the hypertrophic response. Well, according to our data, all the good candidates don't have that big of a role. I think we need to, again, go further in this area. And then one of the last things I'm going to discuss here from the article are future mechanisms that ought to be interrogated. Now, this is not all-inclusive, but one particular topic that has been very prevalent in a lot of different fields is the role of the gut microbiome and in terms of potentially driving the hypertrophic response to resistance training. Again, never waste tissue bank materials. We had tissue sorry, I'm skipping a slide here. One thing related to this topic, we see a lot of reviews in this area, right? There's a lot of speculation. One thing is, hey, look, when you exercise, you may have this change in the biome, right, that produces these magical beans called short-chain fatty acids that leach into the bloodstream and then cause all kinds of crazy effects in tissue, including skeletal muscle. There are a lot of these reviews, but there's not a lot of original data, sadly. And it's just a sad state, because I think there's a lot of speculation in the field without having a lot of these good studies. So to the tissue bank, we had a lot of tissue banked. This was in college age versus older participants, and we had gotten stool from them. We had gotten blood from them. We did DEXAs, we did ultrasounds. We then had these younger and older participants do 10 weeks of resistance training, and we collected the same specimens after that training period. All right, now training worked for everybody. We didn't see an interaction. We just saw a significant training effect where these people gained lean mass according to the DEXA. Well, we took the stool samples and we did biome analysis as well as short-chain fatty acid analysis. We took the blood and we did short-chain fatty acid analysis, both at the pre and post time points. This was our sequencing. We didn't see any changes with resistance training, okay, in terms of biome makeup. And again, apologies for the very small font here, but the first bar, pre and post, is serum acetic acid. We didn't see any change there. By the way, we couldn't detect the other short-chain fatty acids in the blood. So that was the prominent one, and that's the only one we saw. The rest of the bars are the short-chain fatty acids we found in the stool samples. The only thing showing a significant change with training was hexanoic acid. When we ran associations between the changes in these short-chain fatty acids with changes in anabolic outcomes, we saw very, very poor correlations that were nowhere close to being statistically significant. So I think we need to revisit the theme of the gut microbiome and the short-chain fatty acids, curing all diseases on the planet. In particular, I personally don't think that skeletal muscle hypertrophy is related to this. I think it's mechanotransduction based. And so, I'll get off my soapbox. All right. Finally, I'm going to come back to this. And we were very blessed to work with Troy Hornberger. So remember, I said save your receipt, right? Jeff Goldspink was the first mechanistic resistance training study, and he said when you lift weights, at least mice, right, and you see this increase in myofiber size with mechanical overload, this is due to this increase in myofibril size, but not number. Well, I was lucky to run into Troy. We were both interested in this topic, and for those that know Troy, he is a brilliant scientist. He's like, Mike, I think we have a good technique that's not TEM, which is electron microscopy, that can be used to both count and size myofibrils. And we looked at Goldspink's papers, and this is no disrespect to Jeff Goldspink, but when you look at how they sized and counted myofibrils, these were from light micrographs. And if anybody knows, those are really hazy images, especially in 1964. So Troy's like, we gotta revisit this. We gotta revisit this. So I said, okay, we're doing a training study. It's going to be small scale, but we can get you some human muscle. And so this is, again, small apologies, but this is showing you panel A is one myofiber, so one muscle cell, okay? And the box represents a cluster of myofibrils that have been stained for SERCA. So SERCA 1 would be in type 2 myofibers, and SERCA 2 would be in type 1 myofibers. They then used automated analysis to count and size myofibrils. This is called Cell Profiler. It was developed at the Broad Institute, and you can program it in order to do automated analysis. Now, the human study goes like this. We did 7 weeks of resistance training. Again, we had a handful of participants. This is N of 8, but Troy was anxious to get this technique going. You're seeing that training for 7 weeks significantly increased type 2 fiber size, which is a training response that we would expect. This is myofibrils per fiber pre to post. And you see in type 2 fibers, we show an increase. In type 1 fibers, we show no change statistically speaking. And then finally, the size of the myofibrils did not change. So this certainly counters what Goldspink says. And I'm introducing the Hornberger model, which is when you resistance train, this is the first evidence showing that indeed it is an increase in the number of myofibrils versus the size of pre-existing myofibrils. Now, we have a lot of questions, right? Is this because myofibrils split? And Brett, you mentioned, right, we have mitochondria and muscle cells exist as a reticulum. Brian Glancy has great data on this topic. Well, when you look at myofibrils using this 3D SEM, okay, they also exist in a matrix. They don't run parallel to one another. So I think what would be interesting is to see, do we have more branching and splitting with mechanical overload, or do we have myofibrillogenesis, which would be a completely separate mechanism? We're also interested in aging effects, atrophy effects, and related topics as well. And then finally, me personally, I like resistance training, hypertrophy, et cetera, and how different volume loads play into these adaptations. I think that would be really neat to investigate. So what's the talk summary here? Well, to borrow some words from a defense secretary, there are known knowns, right? There are known unknowns, but then there are unknown unknowns, which are the things we do not know we don't know. And that's where I'm at right now. So to the students and young PIs in the room, stay hungry, travel miles and miles before you sleep. My old postdoc mentor told me that, that's Robert Frost, and run through brick walls. I look forward to reading your brilliant discoveries in this area. Thank you.
Thank you, Dr. Roberts, for that great talk. We have time for a few questions, if anyone has any questions. And if there are any questions online, we can take those too, if you just put them in the chat. Any questions for Dr. Roberts?
Okay, so in all of these studies that you guys were summarizing, pulling together, agreeing on, exploring how you disagree in things, when it comes to hypertrophy training, we know that with resistance training, training age has an effect on how fast someone will show hypertrophy. is that kept the same for all of these studies, or is that not even looked at, in a sense, if that makes sense?
I'll go to the microphone. That's a good question. So the question was training age and how that factors in. We collect data via questionnaires. That's the limitation before people train, and we have not seen associations with training age and the training response. Now, that's probably low quality data, right? The question is, what year did you start lifting weights? And then we just do the subtraction from the year 2024. But we also try to gather training frequency and have people expand on that. All that is to say that we're just not seeing a strong correlation there, right? But yeah, I mean, it would be practically impossible to get everybody that's trained for around 10 years and then enroll them, you know. Yeah.
With relation to hypertrophy, when comparing to athletes, you also include a hormonal response. Like with males, the more testosterone you have, the bigger your muscles will get. Not to say you're taking steroids, but comparing two people, depending on where they're at.
Yeah, so we in the review we talk about that. You know, Stu Phillips is on the review, and he certainly has been vocal about the free hormone hypothesis driving the anabolic response. And the reason being because he's looked at the acute response to one bout of exercise, showing that the GH surge or the increase in testosterone doesn't correlate to the long-term training response. Now that's Stu and sort of that mindset there. You know, there's people like Phil Atherton, obviously Bill Kramer, who would sort of counter that perspective. All that is to say, my take is that the protein response in your tissue when you contract that muscle probably has a larger role, right? And I think one very good piece of evidence to support that is when you look at the metadata on men and women, the relative lean mass increase to resistance training is not lower in women, right? The relative increase. Does that make sense? And so I think that certainly supports that the localized response, right? The mechanotransduction response is what drives the hypertrophic response. Yeah. Yep.
Thank you for a great talk.
Thank you.
My question relates to the ribosomal DNA copy
Yeah.
and the lack of a correlation. Could you comment on whether it's the copy number or could those DNA be tagged epigenetically?
Yes. Right, so we've worked with Adam Sharples, who's a pretty smart fella. I always joke, the British accent just grabs you. He sounds really smart. He is smart. You know, he would argue that the epigenetic response is huge. My counter to that is we haven't seen a study where we've used mice to knock out the enzymes that cause methylation or erase methylation and have shown that hypertrophy is impaired. So right now, all of the sort of documented literature is just observation. We have a paper with Adam, right? Most of the methylation to one bout of exercise occurs within a 3-hour period. And that agrees with Julian Zuroth's data, by the way, with one bout of cycling exercise. But there's probably a lot of noise there that doesn't matriculate to mRNA expression and certainly not protein production or translation. So I think it's a little early. I do know Kevin Murak has worked on this in rodents, so you may check out some of his work with methylation to ribosome biogenesis related genes. Yeah.
Hello, Dr. Roberts. First off, excellent presentation. I enjoyed it very well. When you were talking about doing resistance training and achieving muscle hypertrophy at the beginning of your exercise, you had pictures of squats. And so I just wanted to know what was the exact resistance training that was being applied. And I was just wondering if, for the reason being, like I'm trying to see if you can apply this to maybe perhaps an acute population. And so ones who would not be able to do squats at all, would this still translate? If you understand my question.
Yeah, kind of. We can talk a little bit more offline. I mean, I have thoughts about that, but I think it's going to take more than five, like it'll take 20 minutes. The one thing we've gotten accustomed to is doing hex bar deadlifts, but that's from a research perspective. And I tell people all the time, practically, if you go bottom up and lock out on that exercise, you standardize that measurement at pre and post. If you do leg press, you got a lot of herky-jerky bouncing. If you do squat, depth is always an issue. So we do hex bars. Now, if people have lower back issues, right, that could be a consideration. But we can talk about that. Yeah. Long answer. Yeah. Yes, Brett.
Mike, that was a brilliant talk.
Thank you.
So ask something that you really never covered, but I'd like to get your input. Do you think the mechanisms for training-induced hypertrophy and disuse-induced atrophy are completely overlapping, not overlapping?
No, they don't overlap.
Or somewhere in between?
No, they're different. So Stu would tell you that, and we just worked with Marcus on a review there, and that's kind of the premise of the review. So I think muscle protein synthesis would be the common overlay there, and that is to say that with hypertrophy, you would see the increase that's pulsatile in response to each bout. And then with disuse atrophy, and I was shocked, but disuse atrophy, you have a lowering, exponential decrease in protein synthesis. But again, breakdown isn't moving the needle a lot. And there's conflicting data on this point, right? The animal studies would say yes, for sure, but the human studies with chasing tracers would say the breakdown response isn't as impressive as you would think. Does that make sense?
Makes a lot of sense. Does that imply to you then that the interventions to prevent disuse atrophy should be different than interventions to just induce hypertrophy?
Oh, that's going to be another like 20-minute like response.
We can talk later.
Yeah, over a beer especially. That'll get like the gears turning in my head.
Thank you. Any other questions for Dr. Mike Roberts? Yes.
Great job, Mike.
Thank you, Joe. Appreciate it.
It has been a blessing to see you develop over the years. It's unreal. So I recently saw a social media post about stretching.
Yes.
And the mechanical loading associated with for years and years, there's been data on bird wings being stretched in a paper towel roll, right, which is very, very difficult to do in humans, right?
Yep.
But it was apparently tried to replicate.
I think they did.
Yeah.
Can anyone raise their hand if they read that like stretching study where it was quite uncomfortable? It was like 5 minutes of stretches per
3
set,
times a week for some.
it was almost like a weight training, but with stretching.
Pectoralis major, I think, is what they examined.
That's right. Yeah, you saw the picture of them doing this.
Well, I didn't see anything, actually. I just read a title, which is horrible. But talk a little bit about this idea of passive mechanical loading.
Yeah.
And how that could hypertrophy in series versus in parallel.
Oh, gosh.
I don't know.
Joel. Man, y'all are asking things that are going to take 20 minutes. And by the way, I'm no expert in these responses. So I think Troy Hornberger, Kent Jorgensen, and Stu Phillips wrote a review. And what I can tell you, I can send it, right? But they talk about sarcomeres in series with stretching versus radial myofiber hypertrophy. Troy would seem to suggest that the mechanisms are different, and they're starting to get at that. That's all I can tell you. If anyone's listened to Troy Hornberger, you hang on for dear life at conferences when he gives a presentation because you're like, I don't know what you're talking about, man. But he has very sophisticated imaging techniques, and he has a really neat mouse model now that can track the fate of proteins And so he can stretch, he can contract, and he can look at where proteins end up. And I think one of the things he's trying to disentangle is this longitudinal hypertrophy versus this radial hypertrophy. That's about all I can speak to. Yeah. Great question.
All right. Well, big thank you again to Dr. Mike Roberts for
Corey.
your
Yeah,
talk
thank you.
today. Thank you.